In Tanzania, a young female suffering from intestinal parasites chews on the succulent shoots of a small tree called Vernonia amygdalina. Within a day, her symptoms are relieved. Even more interesting is that this is a young female chimpanzee. Now, herbalism is a very appealing notion of using plants in order to treat our symptoms and for health. But what does science really tell us about modern herbalism? The history of herbalism goes back farther than the human species itself. Many animals will chew on different plants when they have symptoms or infections, for example. Every culture has managed, through mostly trial and error, to identify many local plants that have useful medicinal and even recreational purposes. For example, many ancient cultures identified that caraway can be used for indigestion. There are an estimated 400,000 known plant species around the world, likely even more. This is a vast natural chemical laboratory. Now, a lot of the older notions that are bound up with herbalism tend to be mystical in nature, like many beliefs that originated thousands of years ago. The notion that some plants contain a mixture of substances that are specifically designed to be useful medically for people is a mystical one, not one that is in line with modern science. It also tends to be a very anthropocentric view of nature. It is certainly incompatible with an evolutionary view, which tells us that plants, like other things, evolve for their own survival. And there's no particular reason to think that any individual plant will have a special combination of many substances which are designed to be beneficial for human health. Now, despite these mystical roots, it would be silly to ignore the vast potential of science-based herbalism. Again, this vast chemical laboratory out there just waiting for us to explore it. Now, modern herbalism is scientific and has the same kind of relationship to these mystical herbalism as does, say, modern chemistry does to alchemy or modern astronomy does to astrology. We don't ignore or minimize these sciences because of their pre-scientific roots. In fact, the core myth, if you will, of this lecture is that there's something fundamentally different between herbalism and modern pharmacology. In fact, they're all part of the same science, is identifying useful substances that have some biochemical effect in the body that can be exploited. Herbalism, or phytotherapy as it's sometimes called, simply restricts its range to using plant-derived substances rather than anything. Now, there's also a term called pharmacognosy, and that is the study of drugs or drug substances of natural origin, as well as the search for new drugs from natural sources. So a lot of modern pharmacology, in fact, derives from the study of the health effects of plants or things that are derived from plants. Many modern drugs, for example, themselves are plant components, like quinine was originally derived from the cinchona bark. Poppy or opium is a source for morphine and all opiates, which is an important class of pain-relieving drugs. And digitalis was originally purified from foxglove, a pretty flower. I actually have one in my backyard. But digitalis is used for many heart conditions. It basically can affect heart electrical function. And this is still a very active area of research. Now, modern pharmacology, which includes studying plants and other natural sources, has a specific definition. Essentially, a pharmacological agent or drug is any substance that has a biochemical effect on the body, including the microbes in the body, beyond its purely nutritional value. So things that are just nutrients are not drugs by their nature. It alters the biochemistry or function of some or all of the cells in the body. So when herbs are taken to have a biochemical effect on the body, they are being used like drugs. And again, I want to dispel that false dichotomy that something fundamentally different is going on. But how do drugs work? What do we mean by affect the biochemistry of the body? Well, pharmacology is largely an attempt to understand all the different ways that we can interact with the body in order to affect function and health. treat symptoms and alter the course of diseases, for example. Let me run through types of mechanisms of drugs so we can better understand them. One thing that drugs do is they bind to receptors on cells. Now, cells communicate with each other and they read their environment by having chemical signals bind to receptors on or even inside cells. Now, a substance which binds to those receptors and either activates them or blocks or inactivates them is one mechanism, one pharmacological mechanism of some drugs. Drugs may also alter the effects of enzymes. An enzyme is a protein that will make a biochemical reaction go faster. So by interacting with them, a drug may either increase or decrease the rate of a biochemical reaction. Drugs may also activate, inhibit, or mimic the effect of hormones, which are a specific class of substances which do signal throughout the body. They're part of the endocrine system. Drugs may have antimicrobial activity. They may alter the activity of neurotransmitters, which are core to nervous system functioning, so they may affect brain or nerve function by increasing or decreasing the activity of neurotransmitters. They may alter ion channels. Now, these are protein channels on the surface of cells that affect and control the flow of charged ions into and out of the cell and are very important for cell function. They may also alter transcription, the rate at which transfer RNA is transcribed into protein, so it can increase or decrease proteins. There are other mechanisms still. There are osmotic agents which alter the fluid inside the body. They may cause fluid to go from one part of the body or compartment to another. And different other drugs may interfere with cell division or important steps in synthesis, such as chemotherapeutic agents used to treat cancer. And there are still other drugs with other functions. In addition to the actual pharmacological activity that a drug has, we like to know as much about how a drug interacts with the body as possible. They are very important and powerful tools in medicine, so therefore we like to understand them as much as possible. These other aspects of drugs are broken down into two broad categories: pharmacodynamics, which are ways in which the drug affects the body, like many of the things that I just mentioned, and pharmacokinetics. Pharmacokinetics you can think of as the ways in which the body affects the drug. And pharmacokinetics includes things like how the drug is eliminated from the body. Is it through the liver, through the kidney, or some other way? How is it metabolized? Is it detoxified by the liver? Or maybe there are active metabolites. The drug is turned into other drugs that have other functions. We also like to know what the half-life of a drug is. How long does it hang around inside the body? And where does it go? Which parts of the body does it have access to? And is it protein bound? Many substances are bound by proteins in our blood, and protein binding may inactivate part or all of a drug in our blood. Another very important aspect of drugs are bioavailability. This essentially means how much of a substance that we take by a certain route gets into our body and gets to the site where it has its activity. Bioavailability is critical to understanding what the ultimate effect of a drug is going to be in the body. It also determines whether or not a drug could be taken internally, orally, you can eat it, whether it needs to be given intravenously, or whether it can be injected into a muscle or given rectally and absorbed through the rectal mucosa. Those are the common ways in which drugs are given. It also involves how much is metabolized. Now, when you take a drug orally, it gets absorbed in the intestines. The blood that absorbs things from the intestines passes through the liver, through the portal system. Now, this gives the liver an opportunity to get a first crack at any substance which gets absorbed into our system through the intestines. And it may detoxify, it may metabolize a drug before it has a chance to really get distributed to the rest of the body. So even when a drug's getting into the body, the first pass through the liver may inactivate most of it. Essentially, this determines how effective or how potent a drug is going to be in terms of how much of it is going to get to the target tissue. Now, in drug development is largely the process of identifying and isolating very specific active ingredients, a specific molecule. This often does start with identifying the effects of a plant, but then looking at the dozens, sometimes hundreds of individual chemicals or molecules in the parts of that plant, and isolating individual ones and then seeing what their bioavailability is, what their pharmacokinetics are, what their pharmacodynamics are, etc. At that point, chemists may get involved and they may tweak or alter the chemical structure of that molecule in order to increase bioavailability or to make it less toxic, for example. Once the chemists have identified and altered, if necessary, a molecule, then it needs to go into actual testing, which we break down into two basic phases or categories. Preclinical research, which is everything before we give it to people. This includes in vitro studies, in vitro literally translating to in glass, but means in test tubes or petri dishes, and then also in vivo studies in life, studies in animals. Once we've learned as much as we can from in vitro and animal studies, and if a new candidate drug appears to be basically safe and may have some interesting effects that we want to exploit, then we move on to the second type of research called clinical trials. Clinical trials have 4 phases. The first phase is giving small doses to healthy people, again, just to make sure that it doesn't have any unanticipated side effects and to learn more about its pharmacodynamics and pharmacokinetics. Phase 2 trials are small preliminary studies in actual to the target patients, patients with a symptom or a disease that we would be interested in treating. If a drug is safe and shows promise at that point in time, then it goes on to phase 3 trials, which are large, definitive clinical trials. And that's the trials that determine if a drug is going to get approved for the marketplace. Once a drug is on the market, we do what are so-called, or sometimes called, phase 4 studies, which is post-marketing research, where essentially we continue to follow a drug and look for any rare side effects that were not seen during the actual experimental research. So in light of all that, we want to think about what herbs actually are. Now, herbs have pharmacological effects. They have substances which have pharmacokinetics and pharmacodynamics. They are taken so that they can have some kind of biochemical effect similar to what I described. In short, herbs are drugs. But in some countries, like in the United States, for example, they're actually marketed as if they were supplements. They are regulated as food or as if they were vitamins. However, they're generally not taken for their nutritional value. They are taken as pharmacologically active ingredients, drugs. Herbs, in fact, often have many active ingredients because they're not purified. Now, the justification for this is the notion of synergy, that different substances maybe individually don't have much of an effect, but when taken together, they have an important, useful clinical effect. Now, there is, again, a kernel of truth to that, as there often is to misconceptions. There are some times where we would combine 2 or 3 substances together because they have some synergistic quality. Maybe they have the same effect but with different side effects, so we can get the same ultimate clinical effect while minimizing side effects. Or maybe one substance increases the bioavailability of another, the one that actually has the pharmacological effect that we're going for. But these kind of synergistic effects, we only can discover them after doing careful research and combining substances in a way that makes sense. It would be an awful coincidence if they occurred together in the same plant through chance alone. Again, there's no particular reason to think that evolution would select for such combinations. It can still occur. There can be synergistic effects in herbs. However, we can't assume that that's the case. We would hopefully want to base any such determination on actual scientific evidence. It also needs to be noted that these chemical substances, which are taken for their pharmacological activity in plants or herbs, have the same range of side effects and toxicities that any other drugs do. Again, we don't want to fall into the false dichotomy of thinking that herbs are fundamentally different than drugs. Now, what about dosing? One of the primary advantages to the drug development process is that we isolate a specific chemical and then we can deliver it in amounts that are very precisely measured. And we have some idea about what the bioavailability is, how much is going to get into the blood, what the blood levels are going to be, and what the half-life is, how long they're going to stay there. With herbs, we often don't have all or even any of this information. And in fact, when herbs were studied for their likely active ingredients, it turns out that many products have tremendous variations in the amount of active ingredients in various products. For example, a 2003 study of St. John's wort showed a 17-fold difference when looking at markers for how much St. John's wort is actually in the tablet from one pill to the next pill, even in the same bottle. And when one brand is compared to another brand and compared to the labeled dose, the amount that researchers found in the pills in the bottle were varied from 0%, meaning that the label was accurate, to 109%, meaning that there could be twice as much or half as much in the pills as was on the label. So it makes it very challenging to give a precise dose when you don't even know how much of the active ingredients are in the products that you're giving. Now, part of the reason for this variability is that there is a tremendous amount of variation from plant to plant. The amount of different chemicals in a St. John's wort plant, for example, can differ from season to season, from plant to plant, from one part of the plant to another, and from year to year based upon environmental conditions. So it's very hard to control for the amount of active ingredients just by giving a certain amount of the plant itself. There are also issues that come up with quality control. Often there is a lack of standardization, even beyond the notion of dose. Claims for different types of popular herbal remedies are often based upon folklore, sometimes just pure marketing, or at best anecdotal or uncontrolled experience, not hard scientific evidence. Sometimes there are in vitro or, again, preclinical data, which is interesting and may suggest a potential role for an herb, but then those findings are extrapolated to clinical claims. Now, one thing we've learned in the last 100 years or so of doing scientific experiments for medical treatments is that for every 100 or so treatments that look very promising in the laboratory, very few of them actually make it all the way through clinical research and are shown to be safe and effective in people. So it really isn't appropriate to extrapolate from these preclinical studies to clinical claims. There is also the issue of bioavailability. So a substance that's part of an herb or an entire herb may have very interesting effects when you look at what they're doing to cells in a petri dish, but that doesn't mean that those constituents are going to get to those cells in your body. Most drugs, in fact, fail because they lack adequate bioavailability. So any particular plant at random is unlikely statistically to have adequate bioavailability for the putative active ingredients. Recently, it's come to attention also that many herbal products are contaminated. They may have other herbs in them that are not on the label. Sometimes Chinese herbal remedy names may refer to several different plant species, and there have been some cases where the wrong herb was given because of this confusion, leading to significant toxicity. A 2009 study of herbal remedies that were purchased over the internet found that about 20% of them were contaminated with heavy metals above safe limits, including lead, mercury, and arsenic. Some as much as 10,000 times safe limits. So again, this raises the notion that we need better quality control. I'm going to talk about some of the most popular herbal remedies that are on the market and in light of what we have said up to this point. Starting with echinacea. In the middle of the 2000 aughts, if you will, echinacea was the most popular herbal remedy on the market. Echinacea is also called purple coneflower. It comes in various plant species, most commonly promoted for the prevention or the treatment of the common cold. This was first popularized by a German-American, H.C.F. Meyer, in the late 1800s. He claimed he discovered the remedy from the Plains Indians. Now, the Plains Indians did use echinacea for many things, including snake bites and toxicity, but it was never specifically used by them for the common cold. Meyer simply made this up and used the Plains Indians as a popular marketing ploy for the time. Now, of course, American Indians also used many other plants for many reasons. They used tobacco, for example, as an herbal remedy. So the fact that an indigenous population used an herb, at all, let alone for a particular purpose, doesn't mean that necessarily that it would hold up to modern scientific investigation as to its safety or effectiveness, although this does become a very popular marketing strategy. Now, Meyer himself marketed echinacea as a cure-all. He even claimed that it would cure cancer. In Europe is when it really became most popularly recommended for the common cold. But this was not based upon any pre-existing evidence and not based upon the actual history of use of echinacea. But of course, like with all of these claims, the history is interesting and how the claims came about, but at the end of the day, the scientific evidence trumps everything else. Now initially, which is often what we see with herbal remedies and as in many things with medicine, there are many small preliminary studies, and usually the results of such small and preliminary studies are mixed. They're all over the place. Some show benefit, some don't show benefit. But in essence, we're asking, is there any even potential here to justify doing more involved, rigorous, large clinical trials? The largest and best controlled clinical trials that were eventually done on echinacea showed that it had no benefit in the prevention or treatment of the common cold. The biggest one was an NIH study published in the New England Journal of Medicine in 2005. Now, they compared 3 preparations of echinacea versus placebo, a null or an inactive substance, in 437 healthy adults, and they found unequivocally no effect, no benefit from echinacea. Now, those who were selling echinacea for claims that it cured the common cold criticized this research. They said that there are different species of echinacea, and therefore the one that was used in the study wasn't the correct one. They said that different parts of the plant are used, the root, the herb, the flower, or the whole plant, so studying one part doesn't tell you anything about the other parts. There are various preparation methods, dried herbs, extracts, or fresh pressed juice, and therefore, again, testing one doesn't tell you about the others. And that preparations contain combinations of species or plant parts, and not just the purified ones or extracts that were used in the study. However, this amounts to nothing more than special pleading, an excuse for why these studies turned out to be negative. And if what they're saying is true, then how could you know that any particular preparation of echinacea, in fact, worked? What the results of the published research by the NIH did have an effect on the popularity of this herbal remedy. And at one time, it was a $300 million per year market in the U.S. alone, representing about 10% of the herbal market. But after this, it dropped to about half. But that still means, even in the wake of pretty negative evidence, there is still a multi-million-dollar industry for echinacea as an herbal remedy for the common cold. Let's go on to ginkgo biloba, another very common herb that is claimed to have benefit for memory and mental function and maybe even Alzheimer's disease itself, a degenerative form of chronic dementia. The proposed mechanism for ginkgo biloba is that it is a blood thinner. It thins the blood a little bit, and this may lead to improved circulation, including improved blood flow to the brain. However, on close examination, this is not a very plausible mechanism of action. That blood flow to the brain is actually already quite carefully regulated, and unless you have pretty end-stage vascular disease of the blood vessels in your brain, a slight thinning of the blood is not going to improve blood flow and oxygen delivery to your brain. So for most people, it doesn't make any sense that a mild blood thinner would have benefits. In some people with vascular disease, it could make sense. However, if that is the case, we have other blood thinning agents like aspirin, which is much more effective actually than ginkgo biloba. So if ginkgo works by that putative mechanism for dementia, then aspirin should work much better. But as always, we could talk endlessly about mechanism and putative mechanism, but at the end of the day, that needs to be put in the context of actual clinical research. Does it work? Well, a 2008 study published in the Journal of the American Medical Association showed in this study, G. biloba at 120 milligrams twice a day was not effective in reducing either the overall incidence rate of dementia or Alzheimer's disease incidence in elderly individuals with normal cognition or those with minimal cognitive impairment. That's a long way of saying ginkgo biloba doesn't work for memory or dementia. How about St. John's wort, one that I've mentioned previously? This comes from the plant Hypericum porphyratum, and it is claimed to be beneficial for mild depression. The evidence, the clinical evidence, again, shows mixed results, with perhaps some benefit for mild depression, but not for serious depression. A large trial in major depression was negative, but again, the evidence shows that there may be some benefit for mild depression. Now, interestingly, St. John's wort contains several potential active ingredients, including a monoamine oxidase inhibitor. This has already been identified as a class of drugs that is effective for treating depression. So what more evidence is there that St. John's wort is a drug? It actually contains previously identified drugs that are active in depression, so it certainly is plausible that it can have some effectiveness. But is it a good strategy for treating depression? Well, we have all the old problems that I mentioned about dosing and purity. But in addition, because it is a drug, recent evidence has shown us that St. John's wort has all of the drug-drug interactions that you would expect a drug to have. Most notably, it reduces the effectiveness of anti-HIV drugs, a population of patients for which St. John's wort was being specifically recommended to treat mild depression. It also induces enzymes in a liver system that we call the P450 enzyme system. This is a very common enzyme system that metabolizes drugs. When you induce or increase the activity of these enzymes, therefore it can decrease the effectiveness of other drugs that are also metabolized by the P450 system. And St. John's wort may contribute to serotonin syndrome, a side effect of antidepressants. Let's turn now to saw palmetto. This comes from the fruit of the Serenoa repens plant. It is used for benign prostatic hypertrophy or enlargement of the prostate, very common in men. Most men will develop some degree of BPH by the time they get into their 70s. A 2009 Cochrane review of clinical trials concluded Serenoa repens was not more effective than placebo for treatment of urinary symptoms consistent with BPH. So unfortunately there is no effect. Another one, black cohosh, used for symptoms of menopause, hot flashes, mood disturbances, diaphoresis, palpitations, and vaginal dryness. The mechanism is not clear, but we do know that there are no clear estrogen receptor effects. That's the most common mechanism that you would think about. If it does improve those symptoms, and maybe it's working through estrogen receptors, that's been pretty much ruled out. But it may work through some other mechanism. Again, biology is complex. We can't always reach definitive opinions based upon mechanism alone. Now, the clinical data, unfortunately, is scanty. It's only short-term, and the results are mixed. So at this point in time, we simply cannot say if black cohosh is effective in treating symptoms of menopause. But there is special caution in women with a history of breast cancer, because some drugs which are used to treat the same symptoms can increase the risk of recurrence from breast cancer. Therefore, it's reasonable to assume the same precautions until we understand more about this particular herb. So, in conclusion, plants are legitimate sources of safe and effective drugs. Again, it is a very useful natural laboratory of chemicals, and we have already explored a lot of it in order to discover many useful chemicals. Much of the low-hanging fruit, in fact, has already been picked, the very effective drugs that are in common plants that we know about. But there is still a lot of research that can be done, and there may be very effective drugs and treatments and even herbal preparations out there waiting to be discovered by careful research. However, herbal remedies today are poorly regulated. They're mainly regulated as if they were food or as supplements, not as drugs, which they clearly are. In fact, they are drugs that have highly variable dosing and poor quality control. So while I have nothing against the concept of using plant-derived substances or even just straight herbs for health benefits, I think it's important to avoid the false dichotomy of thinking that herbs are acting somehow differently than drugs. I think they should be recognized as the drugs that they are, and regulated appropriately so that they can be used safely and effectively.